Biology, Part 12. Biology, Part 12. Factors which affect enzymes controlled reactions. Enzymes controlled reactions have a number of important characteristics related to the mechanism of enzyme action described above, and to the fact that all enzymes are protein, the effects of temperature, pH, substrate, and enzyme concentration, and those of the inhibitors and cofactors are of particular biological interest and can be summarized as follows. Temperature. The relationship between temperature and reaction rate for a typical enzyme illustrated. The initial increase in the reaction rate with the rising pH has its own effect. When we talk about pH has got a very important factor in determining the fortunes of enzymes. Most enzymes have an optimum pH close to pH 7, which is the normal intracellular pH. This is illustrated in the picture, in the figure. Enzymes which work extracellularly can have very different pH requirement. A notable example is enzymes pepsin, which is found in the stomach and works best in highly acidic conditions in the range pH 1 to 2. pH changes away from optimum can affect enzymes activity adversely in either of the two ways. In cases where the binding or the catalytic sites within the active center take the form of the charged ions, some of these ions are reassociated. The uncharged groups formed as a result can no longer interact with the substrate or the catalytic activity. Substrate concentration and enzyme concentration. The reaction rate of most enzymes controlled reactions varies with the concentration available. Substrate in the way shown in the figure 5.6 also does the same as we assess it and we move along with it. Enzymes inhibitors. Inhibition occurs when the action of enzyme is slowed down by another substance. There are two types of inhibitors: competitive inhibitors and non-competitive inhibitors. Those who are non-practicable inhibitors, they face one kind of repetition there. These substances are chemically closely related to the normal substrate and have a shape which is very similar to when both substrate and inhibitor molecules are present. They compete for the action or the active concern that they may be doing or may be strange for you. Non-competitive inhibitors. The substance are distinguished from competitive inhibitors because they do not combine with the active center of the enzymes and are unaffected substrate concentration. The commonest types include heavy metal ions. The commonest types practical orientation there, and it certainly brings on to, we'll talk about more things on enzyme as I move ahead with Mavidya.